Hello, and welcome to the 3D structure analysis worksheet. We're going to be looking at proteins in the protein data bank from the website rcsb.org. I'm going to walk you guys through the first question, and then from here you should be able to get pretty comfortable with the program and then walk through the rest of the questions yourself. So for the first question, we're asked to search for caffeine 3-RFM. So 3-RFM is the specific protein structure. Since I'm asking you to search for caffeine 3-RFM, that protein interacts with caffeine. So let's go to that protein databank website and we'll start from there. So we are going to rcsb.org. So I'm typing that in the web browser right now. That takes us right to the protein databank website, rcsb.org. For the search, we're asked to search for caffeine 3-RFM. I'm going to just search for 3-RFM. That should get us right where we need to go. That's the first search result. Now, if we were to search for something like caffeine, we could eventually find that, but caffeine actually interacts with a lot of different protein structures. So these are the search results if I search caffeine. So all of these different proteins can potentially interact with caffeine. So we'd have to take a little while to scroll down and find the one that we're looking for, which is this one. So that works. I'll go ahead and select it. Once you select it, you can click on the structure. That will get us most of the information that we need that I walk you through in the first question. Going back to the search results though, if we were to scroll down and look at the structure again, we could go straight to 3D view on that, and that would take us also directly where we need to go. So once we open this, we've got our caffeine structure. Right now I'm highlighted within here, which is the protein structure that we're looking at. I can click on the mouse and drag that around a little bit. But first, let's look at the top here. So this names the compound. It looks like it is a thermostabilized adenosine A2A receptor complex with caffeine. So we've got caffeine in here. And if we move this around in here, we can see a little molecule right there at the end. So right here. So if we zoom in on this with our mouse, this actually is our caffeine molecule that we're looking at right here. So we'll get a couple more close-up views of that in a second. I'm going to actually scroll back out. We can get an overall view of the protein structure here. So on the top here, this gives us the amino acid sequence in this protein structure, going from amino acid 0 all the way through, it looks like if we scroll down, maybe 320 amino acids or so. If we highlight over these, we can actually, scroll down on the page here, if we highlight each of these, on the bottom right-hand corner of this actual mole view that we're looking at, we can actually see the specific amino acid. So right down in this area, once we highlight an amino acid, we can see the name of it. So in this case, I'm on amino acid 227, which is the amino acid lysine. So 227, I know that because right down at the bottom right-hand corner, it says amino acid 227. Also up here, if I go back and scroll over this, it says 221 is alanine. And then if I move over 6 places from there, I'm at 227, and that gets me back over to lysine again, and that's now highlighted. So this is a convenient tool here where we can scroll over these and look at specifically what's highlighted within the structure. You can see that pink highlight is moving across here as I hover over my different one-letter abbreviations for the amino acids to get an idea of exactly where the amino acids are in the structure. All right, so back to the worksheet. Let's double check and see what we need to find. Like I said, we had already selected that we were in mole view, which is the JavaScript viewer. And the next question asks us to describe the difference in styles between cartoon, ball and stick, and label. So let's go back here. Right now, we have this selected here, and nothing is focused. For our components, though, within our components, our polymer is in the cartoon mode. So the polymer is the amino acid chain. Right now I have that highlighted, and specifically all of the amino acids are highlighted. Now, my ligand, my caffeine, was not highlighted there, and now it is. So my ligand is actually in ball and stick mode. So I've actually clicked on that now to zoom in again on the ligand, which is caffeine, which is in ball and stick mode, where my polymer, my amino acids, are in cartoon mode. So in cartoon mode, we actually can't see any of the structures of the amino acids. We can basically just see essentially a ribbon, and a segment of that ribbon represents the amino acid. So let's say we want to add a different representation. So one thing we were asked for is the ball and stick representation. So now all the amino acids are represented by two representations. I'll open that back up again so we can see that one of them is the ball and stick representation that I just selected, and the other is the cartoon representation that was there originally. So the ball and stick hopefully looks familiar because that is how caffeine was originally represented. And you can see I'm highlighting caffeine right here now. It's not connected to any of the chains, but it's a little bit harder to pick that out unless you know exactly where it is, because now we've got so many structures with all our amino acids. So if we take off the cartoon representation, now it even maybe looks more hard to see, but I've got caffeine highlighted again. I'm just going to move this around with my mouse. Caffeine's highlighted. All the amino acids are presented with the ball and stick model that I have selected. All right, let's look at one more option here. So let's add a representation and look at the label option. So I'm going to go ahead and select that. Now label is selected too, which is convenient because now when we zoom in here, if we pick a specific amino acid we want to look at, so right now I have amino acid number 242 selected, which is the phenylalanine. It basically gives us the three-letter abbreviation for that amino acid. So if we zoom back out here, maybe we can try to find caffeine again. It's not super easy to see, and I don't even remember if I'm at the top or the bottom right now. So one good thing to do would be go over and look at our ligand component again, highlight that. That shows me where caffeine is, so right here. So now we can more easily see what amino acids are around caffeine if we're looking at it when we move this around. So these are the different types of labels. So after looking at that, I think that it makes the most sense probably to go back to the original representation, which was the cartoon representation for the amino acids, not show the ball and stick representation. And I don't think labels are all that helpful either, because once we hover over an amino acid, we can see the one-letter label on top or the three-letter abbreviation of the amino acid in the bottom right-hand corner of the MolView viewer. All right, so hopefully you can describe in your own words the differences between those specific styles on the diagram within that program. So for the next question, we're asked to select amino acid 15, and first we want to describe the general location of it in the overall 3RFM structure. So for amino acid 15, just looking at the structure, it looks like an end or starting point is here. And that looks like the starting point since I'm at a pretty low number for my amino acid sequence. This is serine, amino acid 7. The end point then of my specific protein structure is serine, which is amino acid 305. Now I have that highlighted in purple. And we're asked to find amino acid 15. So I can keep going over here and look at all the ones that are highlighted within this ribbon structure here. And I'm at amino acid 17 now, so I went a little bit too far. So right now I'm at 15, and that is alanine. So alanine is amino acid number 15. And looking at the structure, hopefully in class you're close to covering or have already covered the alpha helix structures that have formed. And it looks like within this overall protein structure, that is the vast majority of what we have, is these alpha helix structures. I don't see any beta pleated sheets, which is another common way that amino acids often arrange themselves within a protein structure. We have mostly alpha helix structures. And like we said, since we selected these and we had the number 7 here and we were over 300 here, it looks like of our sequence, for however this is chosen to be numbered, for our sequence, this is the first alpha helix structure, and I would say overall amino acid number 15, alanine, is very early in our first alpha helix structure for this protein complex. Next, we want to name, draw, and number the amino acids covalently bonded to amino acid 15. So going back to the structure here for my cartoon model that I have on here, I might change that in a second, but for alanine, that's amino acid 15. Amino acid 14 is leucine, and amino acid 16 is isoleucine. So we've got leucine, then alanine, then isoleucine. So leucine is covalently bonded to alanine, and alanine is covalently bonded to isoleucine. So I'm actually going to switch this back to the ball and stick representation. Highlight this one more time here. Fortunately, we're not in the middle of it, so we might be able to find where these are again. So here's alanine. Alanine has the R group of a CH3, so that's what my mouse is hovering over right now. That is alanine. Actually, let me show you one more trick here. So if we double click on alanine, amino acid 15, oops, I double clicked twice and then it unselected. It basically zooms in for you and gives you everything around that a little bit more clearly. Specifically, you can focus on it. So I clicked it one more time. It basically shows me all the hydrogen bonding interactions related to this amino acid, alanine, and highlights everything within 5 angstroms. So the distance of 5 angstroms to this specific amino acid. All right, now that we're zoomed in on this, hopefully we agree that the covalently bonded ones are the ones where we have our carbon-nitrogen bonds. So again, this is alanine with my 1 CH3 group right here. Here is my amide functional group. So my carbon double bond oxygen, single bond nitrogen. And this is the amino acid that is covalently bonded to my alanine. So this is 14 and this is leucine. And notice that we have our isobutyl group as our R group. So CH2, CH, and then 2 methyl groups. So we have leucine as one of those amino acids. We can now draw that and hopefully draw the structure. When you draw the structure of the 2 specific amino acids, you can draw them covalently bonded to each other. And then if you want to just end the leucine, so specifically that is an NH2 and pretend it's not bonded to anything, or say it's an R group bonded to the nitrogen, both of those would be fine for the actual worksheet. And then the other amino acid that our amino acid 15 alanine is covalently bonded to is this one right here, which is our isoleucine. So for our isoleucine, amino acid 16, here we have an isobutyl group. So instead of the CH3 group coming off the second to last carbon, it's coming off the first carbon closest to the amino acid backbone. So we have our isobutyl group here. And again, when you draw this structure, definitely show that the nitrogen is bonded to the carbonyl carbon of alanine. But when you go here to draw your carbonyl carbon, if you want to draw that nitrogen with an R group on it, you can. You don't need to keep going in your amino acid chain. You can also draw these 2 amino acids individually to answer this question. So name, draw a number of the amino acids covalently bonded to amino acid 15, which hopefully we agree at this point is alanine. And the two amino acids you're going to draw here are leucine and isoleucine because those are the two amino acids covalently bonded to alanine. All right, next is name, draw a number of the amino acids hydrogen bonded to amino acid 15. So going back to here, we need to reorient ourselves to figure out which one amino acid 15 is. So right here. So when it's highlighted, I can see the specific nitrogen and carbonyl carbon that belong to amino acid 15, which can be confusing when you're looking at a very long sequence of amino acids. So when I highlight this again, this nitrogen right here and this oxygen right here belong to my amino acid alanine. This nitrogen here is bonded to that oxygen of the carbonyl carbon. And it looks like that is amino acid number 11, which is threonine. So threonine here now is highlighted. That has the alcohol functional group on carbon number 2 of the R group. So that is one of the amino acids bonded to alanine, is threonine. And then it looks like the oxygen of the carbonyl carbon on this are bonded to two separate amino acids. So one of them is this one, which is leucine, so amino acid 19. And another is this one, which is valine, which is amino acid 18. So we've got leucine and valine, amino acids 18 and 19 are bonded to alanine. And we also have threonine, which is amino acid 11. So a consistent theme for sure for this is that alanine is bonded to amino acids essentially 4 amino acids away. So number 15 is bonded to number 11, number 15 is bonded to number 19, and there's also an interaction with amino acid number 18. So we've got valine, we've got leucine, and we have threonine as the amino acids this is bonded to. And when I say bonded to, that's the hydrogen bonding portion, so part C of question D. All right, now we're asked to select the caffeine molecule within the 3-RFM structure. And we want to look at the interactions that different caffeine is having with other amino acids. So I'm going to double click again. That unselects everything. I'm going to go back to the cartoon representation, deselect the ball and stick representation of all the amino acids, and we want to zoom in on the caffeine. So I just clicked that and it zoomed in. It gave me my focus surroundings at 5 angstroms in length, everything around it. It shows the different hydrogen bonding interactions as well as other interactions happening with the caffeine. And it shows all the amino acids very close to it. Now, I turned it back to the cartoon representation, but notice that when my focus target is the ball and stick, and for the surroundings, that is selected at ball and stick representation. So it's actually adding in the amino acid structures to it because that is an automatic setting this program currently has when you select a specific compound. When you hyper-focus on a compound, it basically gives you the surroundings and shows you the ball and stick model within the cartoon model for those amino acids. So here again is caffeine. So for our caffeine structure, it looks like the obvious hydrogen bonding interaction is the nitrogen off of this, which definitely has a hydrogen branched on it. So we have our asparagine, and that is the NH. So the hydrogen on asparagine is hydrogen bonding with the oxygen of our caffeine. So this is a ketone functional group on our caffeine, and we have asparagine hydrogen bonding with that. Now that blue line looks remarkably similar to this blue line here, And this is the sulfur of methionine. So this is amino acid number 270, and we have another interaction here. Even though this program says that this is a hydrogen bond between the two, this sulfur is not protonated and this nitrogen is not protonated, so there actually is no hydrogen for the hydrogen bond. So this is actually called a sulfur-nitrogen interaction. So it's a non-covalent bond, and it's definitely a sulfur interaction specifically with nitrogen. So it's a sulfur-nitrogen interaction. This is the non-covalent sulfur interaction between caffeine and amino acid 270 methionine. And on the worksheet, the next interaction we want to look at is the pi system interaction. Looking at the interactions that caffeine has with the different amino acids here, it looks like there are 3 interactions coming from it. We've already determined the hydrogen bonding interaction with the amide functional group of asparagine. We've already looked at the non-covalent sulfur interaction. And now we have our pi system interaction, where the pi system of our phenylalanine, so phenylalanine is amino acid number 168, is interacting with the nitrogen of our caffeine. So those are the last of the interactions. Now on the worksheet for the next question, it actually asks you to draw those interactions. I have it arranged so that caffeine is, I think, looking something like this. So let's go back to the worksheet and double check how I have that arranged. So the nitrogen with the methyl group is up. My carbon double bond oxygen is hydrogen bonding, and I'm showing the asparagine there. So going back to the nitrogen, the methyl group on the nitrogen is up. That's how that is. There's my carbon oxygen double bond. So going back here, it looks like that's all aligned in the same way. So I've already shown the hydrogen bonding interaction. So what you would also want to draw in there is the methionine forming the non-covalent bond with the nitrogen that has a methyl group on the 5-membered ring. So that nitrogen right there with the methyl group on it is forming a non-covalent bond with the sulfur of methionine. So let's go back here. So the nitrogen here with the methyl group on it is forming a non-covalent bond with the sulfur of methionine. So you'd want to draw that in from here. And then for the pi system interaction, that is the nitrogen on the bottom of the 6-membered ring having an interaction with the pi system of phenylalanine. So going back to the worksheet, let's look at these interactions a little bit. I'm actually going to open up ChemDraw to edit this molecule just a little bit so we can show those interactions that I was talking about. So remember, this nitrogen had an interaction with methionine. So we have our sulfur right here, and that's bonded to a methyl group. And then we have a couple CH2s before we get back to our backbone of our amino acid chain. So we've got nitrogen on one side, and we have our carbonyl carbon on the other side. And I didn't draw this stereo-specific, but I don't need much more than this for this specific one. And we'll say we've got an NR group right here, so NR. Now this is interesting. So our sulfur is actually protonated. I'm actually going to take this out. So that sulfur is actually not protonated. It's got a lone pair on it. If it was protonated, it'd have a positive charge. I don't know why it automatically protonated when it had this partial bond to it, but we need to edit this, and this is definitely just our sulfur here. And then, like I said, our aromatic interaction is with the nitrogen on the bottom here. So I'll draw that and then expand. So I'm going to expand this. So this interaction is happening with phenylalanine. Put in my aromatic ring and then scoot it over so that shows it interacting with the middle of it. And then from phenylalanine, we have a methyl group, and then we have a CH2, and that's connected to the backbone of our amino acid chain. So we've got a nitrogen there, and we'll say that's NR, and this is our amide functional group. So I'll put my oxygen there, and we will say that this is also NR. So that's essentially the drawing that I'm looking at, essentially specifying the interactions between our hydrogen bonding interaction between the ketone of our amide functional group and the nitrogen of the other amide functional group of the amino acid asparagine. And we have a pi system interaction between the nitrogen and the aromatic ring of our phenylalanine. And then finally we have the sulfur nitrogen interaction between those two structures there. And for the next question, we want to name and quantify the amino acids forming the 5 angstrom caffeine pocket. So going back to the 3RFM structure here, I've basically zoomed out. We're essentially going to start from scratch. I'm actually just going to hover over caffeine, click it to select it. Again, we have our model of our ligand, which is caffeine, as well as our target area and the surroundings. So essentially the 5 angstrom surrounding area of this. You can again see the interactions this has with our methionine, with our asparagine, with our phenylalanine here. And once I expand the surrounding area, I can actually select it. So I'm going to click on it to select it. That'll keep the full area around it highlighted and keep these highlighted too once I scroll over them. So I can look at essentially everything highlighted here, and it looks like in this top row we've got 3 amino acids. In the next row we've got another 3 amino acids, and in the last row we have 1, 2, 3, 4, 5 amino acids highlighted. So we have a total of 11 amino acids in this pocket. So now you can essentially hover over these, and if we scroll down a little bit, hovering over this, you can look at the 3-letter abbreviation for each amino acid in the bottom right-hand corner, or you can look at the 1-letter abbreviation up here. So I essentially want you to write out the full name of the 11 amino acids that are forming this pocket around caffeine. And this, again, is the 5 angstrom area around caffeine.